Let's see what is there inside. The main thing in my pocket. We'll be counting RBC and WBC. Okay? Which one is for RBC? How do you know? By this, by the red color. Can you see a bulb in the tube?
Yes.
Are there anything within the bulb? A red color bead. And it moves. It's not fixed. Okay. Purpose, I will ask. I will tell first, then we will ask. So, since this is not a WBC counting class, so this is regarding RBC count. So, we'll forget about RBC diluting pipette. So, what is its name?
RBC diluting.
Very smart. She could catch what its name would be. What is the name? RBC diluting pipette. There are 3 identifying marks there. With this we can identify this is a RBC diluting pipette. One thing you said this one and the bed. Okay, second one. And third one is there are certain graduations. Can you see that? One is above the bulb and below. How many divisions are? What are the mark you can get there? You need powerful glass. You are not having. So the boy see the mark.
0.5.
0.5. Another one?
1.
1. Where is 1?
Here.
Here.
And it's
And
101.
101. Fine. Fine. You? Point 5, 0.5, 1, and at the top 101. And here
1.
1. At the middle there is one mark. One is there, one is there. If I take blood up to 0.5 mark, see, it is there. This is the 0.05 mark. If I take blood up to this, then I draw some fluid, RBC diluting fluid, into this. Next, the fluid will come and it will fill this and it will come up to 101. Can you tell me how many times blood has been diluted? I have taken 0.05 up to this, then I sucked some diluting fluid up to this. Then I mixed it. Now blood is being diluted. How many times? And if I take blood instead of 0.5, if I take up to 1, blood is taken up to 1, then if I draw the fluid up to this mark, now it will be diluted, more diluted or less diluted? You have taken blood up to this. Previously you have taken blood up to this. Okay, and during both the times you have drawn up to this. So second time will be more diluted or first time will be more diluted? First time will be more. Okay, very nice.
Why do we dilute this blood for RBC count till such a long extent? While in case of WBC pipette, we only dilute it till 10 times, till the mark is actually 11 in the WBC pipette, we will be showing you later. Because the RBC count in a human body is normally it is 4.5 to 5.5, right? So for the betterment of our counting, we have to dilute it to a large extent so that we can count it and then multiply it by a dilution factor and the volume of the chambers to get the actual result of the patient we are doing RBC count for, right? Then you can detect the RBC pipette. There is a red color bead. I think sir has shown, right? There is a red color bead. There is marking. And in case of a WBC pipette, there is basically a white color bead. The marking is 0.5. You can see, right? The marking is 0.5. Then it's 1. Then it's 11, right? And the dilution is very less compared to RBC count.
101. Then how many times it will be diluted? No. 200 times is, if I take blood for up to this 0.5, then I make diluted it up to 101, then it will be 200 times. Now if I take blood up to this, it will be dilution will be less. Huh? So it will be 100 times. So this will be 100 times. So this way we can make the blood diluted. So if you take 0.5 blood, then you take fluid and up to this mark, then it will be 200 times diluted. If you take this mass, means 1, then draw fluid up to this, then it will be 100 times. Clear? Clear? While drawing blood, you have to draw blood from your fingertip. So first it should be cleaned and it has to be sterile. Okay? Thereafter you will not be touching the tip. You will not be touching. Now you have to prick it. Blood will be using. So you have to draw. In between there should be no air drop. And what happened? Instead of 0.5, blood has came up to this, more than this. No air gap is there. No bubble is there. So we have taken more. So what you will do? With the help of a filter paper or cotton, just touch it. If you hold it, all the blood will be sucked. Okay, just touch it. Then it will be coming. So you hold the tube like this at your eye level, see the mark, and you will touch it. So it will be coming down to this level. So this is first precaution. Whether the required amount of blood you have taken or not. But you cannot make delay, otherwise blood will be coagulated. So you know the coagulation time. So you cannot make more than, you cannot wait more than this time. So before that, you have to make the blood diluted. Clear? Then In a petri dish, there will be fluid. Then you suck the fluid. It has to be filled. Now you have to be very careful. It should not come above 101. So this word is very carefully you have to do. So whatever, even after your careful, your attention, so blood has come to, the fluid has come, or blood is mixed, no, so it will be red in color. It has come up to this. Now what will do? See, here there is no blood. This is actually the fluid. Blood has come here. When I sucked, no, the initial portion blood was there, so it came initially, followed by the fluid. So when you completed this, there are only the fluid. There is no blood. I didn't mix it yet. So by touching it, again you can bring. No, there will not be any change in result. Since this part is only the fluid, no, there is no blood. So I can bring it to the normal mark. Okay? Then my job is mixing. That's why the bead is there. This bead will mix the blood with the fluid. Proper mixing. Done. Okay. You have done this, but there are some fluid here. There you will find there it is not red colored. The blood could not reach here, not diffused here. Okay. So what you will do, just before using this diluted blood, you blow the first drop. So if you draw 1 or 2 drops, so the fluid contained in this part, which are not having proper amount of diluted blood, so that will come out. Then the actual blood which is diluted, that will come. You charge with that fluid, with this mixed diluted blood in the hemocytometer. So these are the things that you have to perform very carefully. One is required amount and proper dilution. Then mixing, and thereafter excluding 1 or 2 drops. This fluid is ready for your, okay. This is actually the hemocytometer. Okay. NEU, BAU, ER. NEU, BAU, ER. Okay. This is the chamber. So this chamber. Now we are not here. You have to pronounce this. We have a particular accent, so we will use that accent since it is very difficult. Okay. Hello, sir.
This is the hemocytometer. This one contains 3 words, 3 different words. Hemo stands for blood, cyto for cell, and meter for measurement or counter. So this is an instrument which is used to count the number of cells which are present in our blood. It includes the Neubauer slide. So the slide which sir has shown you is the Neubauer slide. Then there is a cover slip. When we put the cover slip over the slide, then it becomes the Neubauer chamber. Then there is a RBC pipette and there is a WBC pipette. The Neubauer chamber has a edge-shaped groove. On the horizontal bar which is present in the edge, there are the finely ruled grids which are used for counting the cells. Today we will learn the counting of the RBC cells.
I will be at the same time, I will be demonstrating.
Oh, okay, sir.
This is the picture view,
Yes.
and this is the original view.
Yes.
Okay. Two grids. These are one grid. You will find these are slip. These two sides will act as bridge. Not these two sides. These two sides will be pillar, and this will be the bridge connecting the two pillars. I have to, if I place it connecting both the pillars, now this is a bridge, okay? And you see, we have two grids and the bridge, in between there is a gap. Can you see that? Others see the picture. See the slide. Isn't it? There is a gap. You know how much is this? The height of this gap? So this is 0.1 millimeter. Two tonnage, very good. These are costly only. When we will be determining area, we will see different blocks, small blocks, smaller blocks, large blocks. So they will be having sides. From these sides, we can determine the area of blocks. Okay, small blocks or smaller blocks, the squares, and all the squares, whatever the size is, squares, the height is same, 0.1. So if you want to calculate the volume is, it is square, two sides, same. And if we want to, this is area, this into this is area. And when we multiply area with height, we get volume. So volume here, area, height is fixed. Only side will vary depending on the size of the squares. So with this, by this method, we can calculate the volume also. Okay, and blood will come, diluted blood will enter, will fill this gap. 0.1 mm height जो gap है ना, उसमें क्या रहेगा? Diluted blood. How we will be charging the blood, diluted blood here, we will show it. Next. এটা রাখো তোমার হাতে.
The grids which are present, they are of square shape, and each of them are having a depth of 1/10 mm or 0.1 mm. So I have already told you that if you keep a cover slip over the Neubauer slide, that becomes the Neubauer's chamber. The chamber which is present in our lab is the improved Neubauer's chamber. So there is a difference between the Neubauer's chamber and the improved Neubauer's chamber. This is the Neubauer's chamber, as all of you have seen, and this is the picture with the slide over it, with the cover slip. Each scale is 3 mm wide and 3 mm long, and the whole scale is divided into 9 big squares.
Show it. Now show the square.
These are the 9 big squares. Each one is having a length
From
of
here to
3 mm.
here, how much? What is the distance? 3 mm. This distance? This one? This one? This one? Okay. How many squares? This type of this size, how many are there? 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay. What will be the area? 3 into 3. 3 into 3, 3 millimeter into 3. If you want to know the volume, then 3 into 0.1. Okay, now forget it. Now focus on next part.
Difference table. Difference table. Difference table. You are improved there.
না দরকার নেই, কমপ্লেক্সিটিতে যাব না আগে। হ্যাঁ,
this is the hemocytometer chamber. As you can see, in this there the length is 0.05 mm, and it is the length of the smallest squares. And for the small square, the total length is 0.2 mm.
no, they didn't follow, I think. Yes. How much? Now we are dividing this into 1, 2, 3, 4, 5 parts. So 1 divided by 1 by 5. Okay. Now we are dividing this 1 by 5 area into 1, 2, 3, 4 divisions. Now what will be the side of each division? That is 0.05. Now it is clear to you? Okay? This and this, what is the side? This side 0.05, 0.05 into 0.05 into 0.1, that will be the volume. Area is 0.05 into 0.05. Clear? Clear? Okay. Next.
The central square, as you have seen, is subdivided into.
No, back.
For WBC.
Central square. You are talking about this. How many?
Sir, this central square is having in total 25 small squares, and in between them there are 16 smaller squares. It is central square.
Here the demarcation is not that. In other image, I guess.
It is not conceivable for them.
This one.
This one. Now you see, you concentrate on this area. This area is our point of interest. This area, central area. Okay, this side. Now you see, forget about these small divisions. আগেই ভালো ছিল। Okay.
Sorry.
Wait.
No, এটা তো ল্যাপটপে।
ও ল্যাপটপে, আচ্ছা, এখানে আমার হাত নেই। It was divided into how many parts from here to here? 5. Here to here? Total how many in this area? Sir was telling that. Okay. Now see, this area again we divided. This area is again divided. How many divisions are there? Not 4.
There are 16 divisions.
16. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. 4 into 4? 16. Okay? Now you come to that part. One of this part.
There is a determination টা বলবো ওই কাউন্টিংটা
না এরপরে কি আছে স্লাইডে নেক্সট স্লাইড কি আছে নেক্সট এটা তো বুঝলো হ্যাঁ এই যে এটা হ্যাঁ এটা না ব্যাক 16 no here how many such small square are there in this area? Who said 55? Huh? How many? 25. No. 25 is this area, this type of square. This type of square, there are 25. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. 5, 5, they are 25. But I am talking about this. 16. So 16. How many?
How many smaller squares are there?
How many smaller squares are there?
How many smaller squares are there?
400. One small square, smallest, 80. He 480.
No.
Huh?
480. No, there are 400, but we will count only 80.
There are 480, but we will count how many? See, we will count. There are 16 here. Hello. There are 16 here. We will count. No, we will not count all these cells. We will count this area, and this area. So this area is, consider this area, total area. This is the upper left corner. And this is upper right corner. And this one. And this one. This one. So in these 5 areas, how many smallest division are there? 80. One is having 16 into 5, 80. We will count how many RBCs are there first in the upper right corner, upper left corner, lower left corner, lower right corner, and central. So when we add all these values, then we will get the total RBC present in 80 small divisions. Okay, that is the point. Okay, next. Now we are could we follow this? Understand this? Okay, nice. So we have to count a number of RBC here, number of RBC here, number of RBC here, number of RBC here, and number of RBC here. Next. Okay, they understood this.
Calculation.
এটা না ওইটা
এরপরে স্যার একটা আরেকটা স্লাইড আছে এরপর আরেকটা স্লাইড আছে আর কি কাউন্টের তারপর এটার মধ্যে WBC কাউন্ট
না এটা তো এই নাও can you follow this volume of one small square small
sir this one
volume of one small square
sir this one is for WBC there is another slide.
Okay good this one is automated means you put your blood in a machine, the machine will say there is this number of WBs. You are not performing it, just putting your blood to the counter and this machine will count. This is automated.
Double sound nahi.
Start.
Good afternoon, students. Today I will be discussing the rest of the part, that is methods for counting RBCs. So
in between I will interrupt.
Okay, sir.
So whether this when we will be saying say particular our discussing some particular points. So again I will interfere and ask them whether they could follow these points or not.
So in methods for RBC count, there are usually two methods. Number one is manual method and the second one is automated method. So in manual method, there are four constituents. The first one is Neubauer chamber, the second one is RBC pipette, the third one is RBC diluting fluid, and the fourth one is EDTA sample.
Wait. RBC diluting fluid we have heard, but we don't know the composition. We'll be knowing what is the composition. RBC pipette we have seen, and the chamber, Neubauer chamber we have seen. And the last point, what is the full form of EDTA? Tell. Thalline diamine. Okay. What is this?
That's a complex.
No, this is the complex. What is the purpose of this? This is anticoagulant. But you are taking directly from your fingertip. You are not using any anticoagulant. Sir, you take my blood. I am taking my blood. I will count it later. So what we will do? We will collect some blood in a container, the container containing EDTA. So I will count it later on. So from that blood I will take required amount of blood. So it may be done in the afternoon, in the evening, tomorrow. So at a later stage I can count my RBCs for that purpose. But today or in our class we will not be using it, because we will be directly measuring our own blood. We will not keep it for longer period. Next. So purpose of EDTA, everybody couldn't learn. EDTA full form, write it there. You tell, come to the microphone.
Ethylene diamine.
One word.
Ethylene
Ethylene diamine
tetraacetate.
Okay? You have to remember this. Next.
So the diluting fluid which we are using, the reason for that is we are diluting the RBCs because as we know the RBC count is much more higher than the other cells present in our blood. So and it's highly impossible to count the exact RBCs which are present in our blood. So we are diluting the main blood with the diluting fluid because it also helps in preserving the RBCs, and also it is an isotonic solution, so it will preserve the morphologies of the RBCs.
Okay, wait. Here it is explained why the blood should be diluted. Hello, you please explain why the blood needs to be diluted for counting. Without diluting, if you take the blood directly, and within that you have to count that, so within that area you will find lot of RBCs there. Counting is very difficult. They are closely lying or heaped. Okay, and one may be touching another, overlapping. So in that case, it would be very difficult to count the exact number. So this difficulty may go away if we dilute the blood. So this is the reason. Count is there. How much? 5 millions per cubic millimeter. So it will. So they will be packed in that area. So it will be difficult for us to count. That's why it is impossible. That's why we made it difficult. And while we will be using this the fluid, diluting fluid, it should be isotonic. Why? Why it is necessary to maintain the isotonicity of the fluid? I will ask you. Come here. Come here. You are a smart lady, I know. So, you don't know? Then you have to know. If I place an RBC in a hypertonic solution, in an isotonic solution, in a hypotonic solution, what will happen to the RBCs? Okay, wait. What do you mean by hypertonic?
Hyper high iso
मतलब
हाई कंसंट्रेशन होगा.
This is a fluid. This is a fluid. And it's having a particular osmotic pressure. This is a cell bounded by membrane. Within the cell there is what? Cytoplasm. And this cytoplasm is a fluid. And it is, since there will be the various constituents, different substances, and they will exert a pressure. Okay, this pressure is called osmotic pressure. Okay, I am placing one fluid here. If the osmotic pressure of this is identical to this, then this fluid will be called isotonic to this. The benefit of this, because of presence of this fluid around the RBC, its shape, a morphology will not be changed. Why? Since both of them are having same osmotic pressure, no fluid will be coming from here to here or from here to there. So morphology, size of RBC will remain same. Now see the case. Another solution I kept here. But here the osmotic pressure is lesser than this. Less than this. Osmotic pressure is this, less. Means this is hypotonic. This is hypotonic. Tonic means this osmolality is less. So what will happen? There will be osmosis. There will be osmosis. So fluid from here will enter into this. This is hypotonic. Fluid will enter, osmosis will be there through this membrane. So fluid will come from here to here. As a result, what will happen? And burst. So it will lose its morphology. If its morphology is lost, then how can you calculate the RBCs? Now if you take instead of hypotonic, hypertonic, now what will be the reverse thing will happen? It will draw, it will be dry, it will be squeezed. So again it will lose its morphology. So maintain the normal morphology, the fluid we will be adding to dilute the blood, it should be isotonic. So that fluid should contain a chemical compound which will produce the isotonicity, which will give isotonicity to the fluid. Isotonic to the with reference to RBC. Understood? And for that purpose, what you have to do next? Next, composition. Composition not there? Next. See, this fluid we will be using, or we have shown, it has got a particular name. What is the name? How do you know? How do you know? Oh, you have seen there. Okay. You pronounce Hems fluid. Write down. Hems fluid. And what is the composition of this? Composition. What is the composition of this? Sir, tell.
The composition of Hems fluid, it contains 4 constituents. The first one is sodium chloride, and its composition is 0.5 gram.
0.5 gram in 100 ml. There will be 100 ml water you have taken, and in this you have put 0.5 gram of gram, not milligram. Remember this. I will ask whether it is milligram. You will remember it 0.5, but it is whether kilogram or gram or milligram. I have some doubt. When I see the result. Normal hemoglobin level. 12 to 14 milligram per 100 ml. I gave you example. There's the experience I gathered. It's not milligram, dear. It is gram. That's why you have to remember the units, whether it is milligram or gram. Okay? Another thing. All smart girls, smart boys. I cannot make them full. That's 3 bars. Normal respiratory rate. Sorry? She forgot. Normal respiratory rate. Normal respiratory rate? 1000. Normal respiratory rate? 72 beats per minute. I got the answer. Yes, I know. It is not hard, it is... You cannot do it in 1 minute. Can you continue this? You just think whether it is possible. 72 times breathing per minute, not possible to maintain, sustain. Religion bar I am used to. Our table they will not mention the unit. So you could learn so many things. Okay, you will be learning to. Again, what is the composition of sodium chloride? How much sodium chloride is there? And what is its purpose? It will provide isotonicity. It will provide isotonicity. Okay? Next part is B is
Sodium sulfate.
sodium sulfate. How much? 2.5 gram in 100 ml. Means 2.5 gram percent. And what is the purpose of this? It will act as anticoagulant. It will act as anticoagulant, isn't it?
Yes, sir.
Okay, next.
Mercury chloride.
Mercury chloride. Can you anybody tell what is why it is used here, mercury chloride?
From the back, what I need from book.
Hello? Anybody? No? Okay. Continue.
So the
Remember carefully. I'll ask again.
So the main purpose of
mercury chloride,
is, fluid is to provide antibacterial and antifungal properties.
It is antibacterial.
And
You
antifungal.
will be preserving this. You may require to preserve it, for say few hours. So during this time, there may be antifungal. Some fungus may enter and cause damage of the RBC. Some bacteria may come and damage the RBC. So here it will act as antibacterial, antifungal agent. Moreover, it has another function, fixation, no? Fixation, no. No. Only this is the purpose. So 3 components, and the last component is water. Okay, the first component is used for isotonicity, very good. And the second component as anticoagulant, and the third component. Okay, very nice, wonderful. Next.
So there is another.
No. Now the procedure. Okay, sir will tell, I will show. All the component equipments. Now it is important. To prick, I need spirit. So everything should be there. So then what I will do?
So in the procedure, first we'll assemble all the equipments and then draw our blood directly from the finger after.
So you pricked it,
After sterilizing our fingertips.
or there will be some blood. It is not yours, it may be your patient's blood. So from there you will draw. Or if it is your own blood, you will be collecting it from your fingertip.
The first drop we must discard it.
Why?
Then
Gram? Milligram? Why first drop should be removed? Excluded? Yeah? Oh, it may cause infection. First drop. No, re bhaiya. Blood. When first drop may contain tissue fluid. Okay, so that's why I have rejected it. Clear? Next. কত রকম কনসেপ্ট আসবে।
So after that, with the help of the RBC pipette, we will suck our blood
Up
with
to?
the help of mouthpiece.
If it is more? Okay, very good. Next?
So if it is more than 0.5, so we need to discard, and the margin should be exact.
Exactly.
The 0.5.
If you take more,
not less.
your count will be more than actual. If you take less, it will be less than actual.
So after that, we will be taking the dilution fluid.
This point, second point, I didn't tell you. From the tip, extra blood should be removed.
And then we'll take the dilution fluid till 0.1 mark.
Easy. Now, next part, you have taken
Sorry,
the blood,
100
and there is the
101.
fluid in petri dish. Now you will be sucking fluid up to this. If it is more than this, better it will be. You cannot control, I will draw up to this. It may go beyond this. So in that case, like the previous one method, you just bring it down to the normal. If it is less, again you have to draw.
So after that, we will keep the RBC pipette in our palms and rotate it gently for 2 to 3 minutes.
2 to 3 minutes.
So that it gets mixed properly with the diluting fluid, and this dilution will be 1:200.
So here blood is one part and fluid is
200.
200 part. So it is 1:200. Dilution is 1:200. Then this one, chamber. Now, before this, it is ready, no? Okay, you keep it. Then, hello, I'll sit there. Go. I'll sit here. This is the hemocytometer. So, it is done. Now I place it under the microscope. So I placed it, then I'll place the cover slip, connecting the two pillars. So, cover slip. It is, I am not doing it on my hand. It is there. So connect both the breeze. You decide which grid you will be using. There will be two grids, no? Upper one, lower one. Better you use the upper one. Why it is better? Because charging will be easier for me from that side. What is the tube? With blood? Okay. So now this is, we have placed it. Now I have to charge the fluid. Okay. Again here, it should be at, whatever. Anything wrong? You demonstrate. Huh? So why you are laughing? Whatever, tell me. Tell. Okay. So now you will charge like this. You touch it, or by simply touching, it will come automatically. Fluid will come. Okay. No, blow korte hobe. If you touch, automatically fluid will be coming by the capillary action. At the bottom there is a gap, no? So it will suck. Automatically it will suck the fluid. And it will come, it will fill the gap. So charging is over. Charging is over. Cover slip is there. Now you are to focus. Before that, you have focused. Before that, you have focused the grids for identifying those areas. There are some area I have selected. I will be counting these and these 5 areas. So you focus those areas. It should be visible to you. Okay. Now you see whether those, after charging, whether you can find those lines. If it is possible, then you will be able to count them. And what is the will be the power? 40x. No, 10x.
First you will see at 10x.
Yeah. So that for focusing, first you will be focusing at 10x. That part we will teach you later. How to focus. That part we will be describing later.
Sir, the blood from the pipette, where will touch that mouth on that display? After putting the cover slip,
it should come within the space. There is a narrow gap, no? It should come. So this tip of this should be close to this. That gap. Then only fluid will come.
so if the cover slip
At the bottom,
if
it is the gap is at the bottom of this. I will not press over the bottom or over the surface. At the gap.
so sir, if the cover slip it slips,
No, it will not slip. It is relatively thicker. This cover slip is not like ordinary cover slip. It is relatively thicker. এই জায়গাটা ঠিকই বলেছো, ওকে একটু বুঝিয়ে দিই। একটা কভার স্লিপ এটা তো আবার ইয়ে হয়ে গেল। 1 2 3 4 5. So my interest is this one. My point of interest is this one and the center one, and this I am unable to see, but these are there.
So they can see here, no?
Yes, I'll show this. You can see there. So I have to count the RBCs. How many RBCs are here? 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 And how many RBCs are? আচ্ছা থাক হলো। Now let us count. Let us count the center part. Okay. So this is our center part. This part we are going to number of RBCs. How many RBCs are there? We are going to calculate. Count. I have told you there is. You will be following only one rule. I will count left side any RBC that are touching the left side and the RBCs that are touching the lower side is my property. Means this is the center part. The RBCs who will be touching these, they will be my property. And those are in between, they are also my property. You will find within this lot of RBCs. Let's first see how many RBCs are here. So here I found, see, your boundary is the middle one. You forget about these two lines. There are three lines, no? So you forget about the right and left side lines. Only the center line is your main line. Okay? Similarly, this is your line of interest. This is also your line of interest. This is your line of interest. Like here also, this is your line of interest. Okay? Now, let us go for this. How many RBCs are there? There. If I find an RBC is touching just this line, it belongs to this smallest division. And another RBC may be here, it is obviously under this smallest square. Now, if any RBC touches this line is not mine. This line touching RBC, the RBC touching this line will be the property of this. My property will be touching this line. The RBCs will be, the RBCs touching this line, my property, and RBC touching the middle one is my property beside this. But those who are touching this line and this, they are not my property. Is that clear? Now see how many RBCs you have. You count. So there is one RBC. One RBC is like this, not touching this one. Another RBC is just touching this one. Main part is here. Even then it is my property. Because you touched my middle line. Because you touched my middle line. And see another RBC. Another RBC just touch this line, but it is there inside. Most part is inside there, but it is touching. But it is not the property of mine. It is the property because this line is not mine. Understood? So this is very important. Which we will be considering you belong. So this way, you jump, it's over, now you come to this. The RBCs you touching there, you have excluded them, now they will be included. Previously you have excluded them because it has touched the right side border. Now it is, when you jump from this side to this side, now this line is belong to you. This side belongs to you. Now any RBC touching this line, I will count for this square, and any RBC within this, it is my property, but any RBC touching this middle line is not my property. Any RBC touching this line is not my property, but any RBC touching this line, it is the property of this. Is that clear to everybody? Any doubt? If you have a doubt, you can talk to me. You can tell me.
Please tell it again.
Huh?
Can you please tell it again?
Which one?
The RBC touching up and
You are counting this line, na? Total number of RBC you will be counting. How many RBCs are there? So these are containing small divisions. So first, I will consider this one. Let's see how many RBCs are there. So while counting this, I will consider these lines. This is not my lines. My line is this one and this one. These two lines are not mine. Any RBC touching this line will be excluded while counting number of RBC here. So I will be considering those RBCs which are within this and which are touching this line and this line. Now clear? So when it is completed, I am shifting. Now I am shifting. It is completed. Now I am shifting from here to there, here. Now I will be counting number of RBCs here. So now this is my left and this is my lower. So any RBC touching these two lines I will count, and which are free here, this I will count. But any RBC touching this line and this line I will not count. Same principle. So when it is completed, then I will move to this. Same principle I will add up. Then I will come to this. Same principle I add up. I will not be counting any cell here. I will not be counting any touching this line. Here I will not be counting. Okay? The top 4 smallest box, number of RBC in 4 top boxes I have counted. Now I will shift. It is completed, now I will shift here. Following the same principle, I will be counting them. Now top, previously I have counted them. Now I will not I will not be counting them. I will be counting them. Okay, same principle I will be following here. When this is completed, I will come down here. When it is completed, Then I will again complete here. I will be coming down here. Then I will finish it. Is that clear to you now?
So it is shown in the next slide. The zigzag pattern. The zigzag pattern is shown in the next slide.
Show the next slide. Next slide. From left to right, movement. From left to right, completed, come down. Then from right to left, completed, then come down, again left to right, come down, again right to left. So this is zigzag pattern. So we have to follow zigzag pattern. Anything else? Okay, এবারে আমরা এক্সারসাইজটা দিই, কালকের এক্সারসাইজটা, দুটো বুক নাও, দুটো বই। হ্যাঁ, ওই যে কাউন্টিংটা। হ্যাঁ, see, this is what the central part. So we have completed the central part, but other four is remained. Other four are remained. Top left, top right, bottom left, bottom right. Only the center part is counted. So I will finish them. So count it. This plus this plus this plus this plus this. How many? Say 120. Say 120. Right. $120 RBC total. My box A, B plus B plus C plus D plus E means upper right, upper left plus upper right plus central plus lower left plus lower right. Total how many? 120. Suppose it is 120. Now can you tell me how many smallest divisions are there in these 580? So 80 divisions contain 120. So that you have to remember. Now you go through the calculation. Oh, see that zigzag pattern. They will be confused. Don't look at this. Don't look at this. Move. Otherwise, this will make confusion. Calculation. This one. Let the number of RBCs counted in 80 small squares. Number is n number. Here, how many you got? And here how many you got in 80 divisions? 120. So your example is 120. Okay. Then number of RBC in 1/50 cubic millimeter. It means it is number is n. Okay. Calculation ta kodai korlo?
Actually here this is the small box. Okay, so we are counting according to the small box. So in one box, if we count the area, so it will be 1/5. So as there was 5 boxes, small boxes. So if you calculate, then it will come 1/5.
Volume of one small box.
Yes.
Volume of one small box, 1/5 into 1/5 into 0.01. So you have total 5 number of boxes. So what will be the result? 1/50.
What is 1 means 1/10. So 5 and 5 will cancel. It will be 1/5 will cancel. So 1/50.
Number of RBCs in this area is 9 and 120. Number of RBCs in 1 cubic millimeter.
So it's unitary method of calculation.
Just use unitary method. In this area, there are 120. How many will be there in 1 cubic millimeter? in 1 by 50th millimeter cubic millimeter, there is 120 number of RBCs. So how many will be there in 1 cubic millimeter? 120 into 50. How much it comes? Are you, what is the result? 120 into 50? 6,000. 6,000. You have diluted blood 200 times. How many times? 200. So you have to multiply this value with 200. Then you will be getting the actually original count that is present in, not diluted, present in original blood of 1 cubic millimeter. So how much it comes? 6,000 into 200.
Result.
Result?
12 lakhs what? 12 lakh rupees,
12 lakhs in 1 cubic meter or 1 cubic millimeter or 1 cubic centimeter. That you have to tell. Every time I insist you to tell the unit, isn't it? So here your unit is cubic millimeter.
So there may be some
Technical error.
technical errors.
You have to avoid this error.
Like if technical errors can be minimized by like if our apparatus is defective, and if we want to minimize it, so the it should be calibrated accurately by carefully by the technique, and there may be some inherent errors, like the distribution of the RBCs they have in the chambers are not visualized, and these things may be there.
You forget about technical errors, point 1, initial one. You forget. They are telling what your apparatus, nowadays it is written made in Germany, but actually these are made in some other place, maybe China, maybe in India, and embossed made in Germany. So it may be the faulty. I have seen some Neubauer chamber. When it is purchased, when I have examined under microscope, I don't find any grids, any such lines. And if the lines are present, these are not correct. So these are faulty equipment, faulty instruments. So because of these faulty instruments, the error may come. So forget, we are sure that instruments are not faulty. So forget about the initial technical error, and what is written there in inherent error. What is that written at the bottom? Here I will be able to read.
Counting a large number of RBCs.
এটা স্লাইডটা ওই জায়গাতে যাও. This is of an irregular pattern in the counting chamber. This affects this may be a faulty pattern. This affects the accuracy of visual count, which can be minimized by counting a large number of RBCs. Means what? You will find you have given 5 areas, no? In one area you will find number of RBCs less. In another, you will find area, more RBCs are there. So, the distribution is not equal. That's why we have taken 5 areas. If I take only one area, this will give 40 result. But where there is dense, if I take this only, then number will be more. If I count 5 areas, then average it. What you did? 120. Total sum is 120 divided by 80 means average per small area. So I have averaged it. So in this way, this error can be minimized. This is what it has been said there, inherent error. That's why 5 blocks we have to calculate. We have to count the number of RBCs in 5 blocks. এটাই তো পয়েন্টটা না নেক্সট এদিকে না কার্সারটা এই সাইডে আনো লেফটে আনো পরে নিচের দিকে আসো কার্সার এইদিকে আনো এখানে হ্যাঁ এবার এটা ট্যাপ করো হ্যাঁ এবার নিচে আসো বাস অ্যারোকে okay sir, this is the normal count, normal range. So note it down. We will be asking by Babusi question regarding this. Okay, now he will summarize the procedure. Procedure. Summarize it.
Are we doing this practical? To count our RBC. It can be counted by 2 methods, visual method or automated methods. This is visual method. We are going to count it by ourselves. Okay, automated method is done by automated analyzer or machine. Okay. At first, what we are doing, like every other hematology practicals, we are going to need to give a prick to these 3 fingers. There was a question why these 3 fingers are pricked, and the first drop needs to be wiped out. Why needs to be wiped out? Tissue fluid. Okay, then we are going to take the next drop of blood into that RBC pipette. Till how much? Till 0.5 marks. Okay, it's not 0.5 microliter or ml. It's just a mark. Okay, it's just a calibration mark. Okay, till 0.5 we will take blood. Then what we will do? Will you wait for more than 2 minutes, 3 minutes, or immediately you will go for dilution fluid? Why we need to dilute that fluid or need to dilute our blood? Yes? We need to dilute our blood because the concentration of RBC is very high. It's around 4.5 to 5.5 million. So if you are going to count it in a chamber, you will see so much crowding in the chamber, and it will be very difficult for you to count it. Okay, if in a small box there are hundreds of RBC, it will be difficult for you to count accurately. So we need to dilute it. So what is the dilution factor here? 200 times. How is it 200 times? See, okay, we will come to that. At first, we have taken that blood, our blood sample or blood drop till 0.5. Then we are drawing or sucking that RBC diluting fluid, which is called Hems fluid. Hems fluid has different constituents like NaCl, sodium chloride. What is its purpose? It maintains the isotonicity so that the RBC does not lyse or doesn't shrink. Okay, so that it is isoosmolar. And what is the function of the next sodium sulfate? Only one guy. Actually, sir, it's not anticoagulant. It helps in preventing Role formation or aggregation of RBC. Okay, sodium sulfate
is used because it prevents the Rayleigh formation. You know, RBC has biconcave shape. One RBC can pile up over another. So if we can see one RBC, but there will be multiple RBC Rayleigh formation. Okay. So to prevent that, we use sodium sulfate. And what is the purpose of this mercury chloride? Antibacterial and antifungal, or it is used as a disinfectant, so it is not infected by other microorganisms. And distilled water is taken like 100 ml. So from that Heim's fluid we are going to suck that Heim's fluid till which mark? 101. So the blood and the fluid is going to be mixed in the bulb area. The bulge area, it's called the bulb area. The blood and the Heim's fluid or the diluting fluid will not be mixing in that tube area. It will be mixing only in the bulb area. So what are you doing next? We need to roll it up. Why? For proper mixing of the RBCs with the diluting fluid. Okay, we are mixing it for how many minutes? 2 to 3 minutes. So after 2 to 3 minutes there is proper mixing of RBC with diluting fluid. Okay. Now we discard 2 to 3 drops of that solution before charging our Neubauer chamber. Why?
So as to get that mixed blood.
Yes, you are quite right. We need to discard the first 2 to 3 drops because that I told you now that a pipette area is not responsible for mixing of the RBCs with or blood with diluting fluid. The blood and the diluting fluid mixes only in the bulb area. So that the last portion here, the fluid remaining here from here to here, it will not be mixed with blood. The entire mixing is here. So we need to discard this part because if you are going to use this part, what will happen? This part is only diluting fluid.
You will not find
Any RBC here. So you are discarding this part. Now here comes how it is 200 times dilution. The entire dilution happens here. Entire dilution happens in the bulb. What is the area of the bulb? See, 101 minus 1. कितना हुआ? 100 parts. Okay, इसमें कितना part blood गया? 0.5. So out of 100, 0.5 is blood. And what are the rest part is diluting fluid, 99.5. So if you are going to see the portion of diluting fluid here is 1:200. Got it? Out of 100 it is 99.5. So out of 200 it's around 1:200. अब समझ में आया? Okay. Now after 2 to 3 minutes we are going to charge the Neubauer chamber. Which what is the angle? 45 degree. Are you going to touch it or just the drop will touch it? Will touch the cover slip. Okay. Will you use the cover slip first or the drop first? Cover slip first, and then you are going to touch the drop near the cover slip, and it will charge automatically. You can charge twice. Okay, there are 2 chambers, upper and lower. You can charge both or you can charge one. If you charge both, it's better for you. And next we are going to count. Okay, counting by zigzag manner. How many chamber are you counting? So how many boxes are you counting?
5 boxes.
Mainly in the central big box, we are counting the 5 boxes. What are the 5 boxes? Left upper, right upper, left lower, right lower, and the central part. Okay, and we are counting only the cells that are touching the left line and the lower line. Okay. If a cell is touching the upper line, will you count it?
No.
What is the line here in consideration? There are 3 lines now in Neubauer chamber. You have seen there are 3 lines. Middle one. The middle one is the line in consideration. If it is touching the middle one, then you will consider it. Okay. Okay. So now we are coming to the calculation part. Before that, every small square has 16 smallest square. Okay. So in total we are counting 80 smallest square. Now we are coming to the calculation where we are going to count the volume of the blood here and the number of cells we have counted. So by that we are going to count like how many number of cells are there in that particular volume, and then we are going to see how many number of cells are there in per cubic millimeter of blood. Okay. Now see, suppose the n value, that means we have calculated around 500 cells. Okay, we have calculated around 500 cells. So what is the volume that for which we have calculated? We have seen now the calculation 1 by 5 into 1 by 5 into 0.1 into 5. 1 by 5 is the length. You know now the area, surface area of square is length square, length into length. L square, length into length, that means 0.5, 1 by 5 into 1 by 5, means 1 by 25. And what is the formula of volume? Surface area into height. Height is 0.1. So you have calculated 1 by 25 into 0.1. 0.1 means 1 by 10. Okay. And then how many number of square we are counting? 5 into 5. So the volume comes around 1 by 50. Okay, and regarding that we have seen the total count can be calculated by n into 10,000 at the end. Okay, so if the n number is 500, what will be the calculation? 5 lakhs. You know, Sir has told that at that time that was 120 and your calculation came out to be 120, so 1.2 million. Sorry, 50 lakhs, yeah. Then if it is 500, it will come 5 million, means 50 lakhs, which is the average number of RBCs seen in blood per cubic millimeter. So that is the entire purpose of this practical, to calculate the value of RBC. Okay, thank you.
To tissue.
But this is the only function?
Then it will carry hemoglobin to the tissue.
It is having, obviously it is having hemoglobin, that's why it can carry oxygen. So any other function? Okay, you don't have any other stock. Who can tell? You do come. Remember what he is telling.
The RBC rupture can gives heme and the
No, functions of RBC. What is function? What are the functions? Tell them.
RBC's main function is to transport oxygen from the pulmonary system to the tissues and cells, and to transport the carbon dioxide and other waste materials from the cells to the pulmonary system for release.
Not only oxygen, it will also carry. How it can carry carbon dioxide?
In
RBC?
form.
How? Carboxyhemoglobin. Carbaminohemoglobin. Okay. Within the RBC, there is hemoglobin. So it may form a chemical compound with carbon dioxide and form a compound. Okay. Beside this, the cytoplasm of the RBC, it can also carry some part of carbon dioxide as bicarbonate ion. Carbon dioxide, RBC will contain a lot of Ca, carbonic anhydrase. So carbon dioxide entering in RBC, it will immediately combine with water and will form hydrogen ion plus bicarbonate. So carbon dioxide actually transferred into bicarbonate ion. So now this bicarbonate from RBC, some part will be retained within the RBC cytoplasm and some will enter into the plasma. And this way, this carbon dioxide will be carried from tissue level to pulmonary system. So this is another function, means respiratory function, carrying oxygen from lung to the tissues, carrying carbon dioxide from the tissues to the lung. Beside it, it has other function.
Now see what is the dilution?
1:200.
0.5 is in 100. 1 will be in 200. Simple. A content of blood is 0.5 in 100. And then 1 will be in 200. So we calculate like this: 0.5 put in numerator, take 1 So it's 1 in 200. So the dilution is 200. It is 200 times diluted. জানবো এটা কম আসবে। Now we'll multiply by 200 and we'll get that. Got it? कितना hard है? Now your job is to identify the number of cells, not to confuse like which one to consider, which one not to consider. Okay. Now, can you differentiate between WBC pipette and RBC pipette? White bead. Only white bead. And the marks.
नहीं
There are different calibrations. The beads. Okay. Bead is for visual. Like you will see from far and okay. The RBC one is the WBC one. Okay. So for that purpose, you are sure now RBC and WBC. Second one, marking. It will be for RBC, it will be 0.5, 1, 100. For WBC, it will be 0.5, 1, 11. Why 11? Why are we diluting it less? Because the content of WBC is lesser now. We could dilute RBC more because its concentration is so high. Diluting more, you can see the RBC cells here. But WBC cells require 101. इसमें 100 ब्लड जो है ये पुश होके इधर चला गया मतलब इतना कर लेंगे ना फ्लूइड तभी तो इधर अच्छा ब्लड अंदर चला गया अच्छा फ्लूइड भी अंदर चला गया ठीक है वाइट एक्स्ट्रा फ्लूइड सेल दो There may be a chance of WBC in that diversion. So you can possibly get WBC something, but we are not taking WBC. Why? Why we don't count or don't take into consideration the WBC count while we are getting RBC in diversion? Just try to find it. या तो ऐसे